Hello once again. Welcome to Grumpy Science. I'm Bob Dennis, and this is Mark Tommerdahl, Grumpy Scientists.
You know, our goal is to be the Wayne's World of the neuroscience community, but you know, we don't have any music in the background. So
not really musical.
Anyway, we want to follow up from the last video, which was talking about methods. And in general, you know, if somebody were to dive into all the publications that they were looking at, you say, wow, you know, this method, and we can actually talk about the methods because we listed them. They're still in our media center. I'm going to pull this down.
Our multimedia command and control center.
Yeah. And basically we listed a whole bunch of methods that you see in the literature a lot. And people that have those methods swear by them, and they say, oh, this is the best method for this, and they're going to keep doing that. Just remember, whenever you hear any of that, including when you're listening to us, that when you have a hammer, the world is full of nails. And
that's the way it looks. Scientists are no exception. People use the tools they have. They use the things they want to use.
Now, we did spend 14, 15 years developing a particular tool. And before that, I spent 20, 30 years trying to study how to develop that tool. And the target was coming up with a generalized tool for brain health. And basically, one of the things that we knew we could study were dynamics and how the cortex changes and is plastic on different time scales. So basically what that means is, what is plasticity? So plasticity or learning is the fact that every single thing that you do in the world has an effect on your brain. It all comes back to haunt you. And it basically leads to rewiring. And do you know when first person that I know of that made a statement about plasticity was? Who the first person to? Now, a lot of people are taking credit for it in the 1990s and 2000 with a lot of different kind of brain games that I'm not.
I'm about to learn something. I'm kind of into science history and. Okay, so enlighten me.
So who would come up with the first idea about a thing called plasticity, where neurons change their properties? Do you think it was like somebody who had their hands inside a brain and looking at it all the time?
Well, I seem to recall, and having recently read a biography of Leonardo da Vinci, that he did some of the finest anatomical studies, and he did do some correlations between brain structure and what people did.
Okay, you're going too far back.
I'm not too far back.
I'm not as scholarly as Bob, so I only going back to the 1950s. Right. Guy named Hebb, H-E-B-B, discovered or proposed Hebbian plasticity, and that was whenever a neuron or synapse was active, it became stronger. So you have pathways in the brain that keep becoming stronger and stronger and stronger. And like I said, a lot of people try to take credit for this years later.
So this is what, 1950s?
Yeah, 1950s. So that was, and you guys, you can comment and correct me if I'm wrong, but I'm pretty sure that's when it was. Anyway, this guy proposed that, and then there were a lot of other studies, and a lot of interesting studies later, where people were doing recordings from repeated stimuli. And repeating stimuli, what I'm talking about is the kind of stimuli that we deliver that are repetitive are sinusoidal. You want to draw a sinusoid?
Yeah.
Where'd the pen go? Here's a pen.
Hold on.
This is our Bob never draws up here. My handwriting's obviously not good.
He's tired of me making fun of him. Sinusoid is a smooth curve that goes up and down like this. Right?
So this sinusoid is actually a very good experimental method. I mean, it gives you a very good experimental tool. So if you deliver a mechanical sinusoid to one place on the skin, it activates the brain, and neurons fire when it goes into the skin, and they quit firing when it goes out of the skin. And the first round, the first sinusoid where neurons respond, it's really, really messy. And the second sinusoid, it gets a little cleaner. And the third and the fourth and the fifth sinusoid get really, really clean, to where at the first, where you had a neuron firing a whole bunch of times on this, it's like, man, I've seen this before. And it's going to fire once, and it might fire again. Maybe not. But by the end, it gets really tight. But two things happen. One, what's called overall mean firing rate. The firing rate response of the sinusoid to that sinusoid goes down. But the entrainment, how well it fits that sinusoid, gets better. So your brain gets sharper and sharper with that stimulus, and what that tells you is, oh, that's really good.
Now let's talk about this a little bit. That stimulus is a sinusoid that's happening at several hertz, right?
Yeah, and usually we deliver the stuff that we
that
deliver
means
here
sinusoidal
on here
cycles per second.
Yeah.
So roughly how many hertz, right?
25 hertz is what the stimulus
25.
on these
25 cycles. That's one cycle right there, about
Yeah.
a little bit more than one.
So this
per
is
second.
1/25th of a second,
Right.
even though it took Bob a lot longer to draw it.
That's right.
But that's
I was trying to be
1/25th
really
of a
careful.
second.
My graphic skills. So
Yeah.
what I would like to point out here is that what Mark is talking about is that the brain is dynamically adapting to this very quick stimulus.
Yeah,
I think that's where you're
absolutely.
headed, right? So
Absolutely. And so if you deliver
each cycle, it gets
bunch of
better
cycles,
at responding, and it's doing that 25 times a second in this case.
and so what people would do is they deliver a whole bunch of cycles, and then they wait a while, and they deliver a whole bunch of cycles again, and guess what? The pattern was never quite exactly the same. A lot of people would ascribe this to experimental sloppiness. Actually,
Right.
my mentor described it as interesting, and actually which receptor systems had a lot to do with that difference. And we actually studied the difference and the history of that pattern, and how did that pattern change with time. And there were a lot of different ways to study it, but one way to talk about is, let's compare it to, well, we can't really compare it to these methods. Oh,
No.
how about that? If you really want to study this plasticity at very high resolution, you got to do something at fairly high resolution. And that's kind of redundant, but which of these methods would be capable of studying something on that
Ooh, me.
yeah, what do you think?
Pick me.
Oh, wait a minute.
Anyone Bueller?
There's somebody over there in the back.
No, me,
Okay.
me. I will take a guess.
He always
This is
raises
a
his hand
guess.
in class.
I know, I'm the worst. Even in class I won.
I sat in the
think
back and didn't
the only
answer
one that
it.
has the temporal resolution that can tell you something maybe that's happening many, many times a second is probably EEG. Am I right?
Possibly,
No,
but
well, possibly. I could be right.
okay, sure. But you got electrodes on outside the
Yeah, I
skull
know.
on this.
I know.
and it's
I
kind
didn't
of.
want to say that.
But
one.
you're right. EEG could do it, and if you deliver a really big stimulus like this, and then another pulse a little while later. We won't get into which time and all that, because different times mean different things. And then you measure the response. Let's say this is the input, stimulus. Now let's look at the output, and say the output likes that, and we deliver another stimulus. What do you think that output's going to look like? Is it going to look like this? Is it going to be bigger or smaller?
It's probably going to be a little smaller.
It's going to be a little smaller.
Yeah.
It's called paired pulse stimulation.
Right.
And so basically you deliver two stimuli and record with EEG, and you got to do it at the right sequence and get it all right, and you got to have the electrodes on just the right place. But you can see this paired pulse sequence.
Right.
Now guess what? What happens? What do you think? What kind of people do you think you see a response like that on? Where
I know the
the
answer to
second
this.
response is the same as the first response.
So talk a little bit about what's happening there, right? So if you've got the same stimulus, we'll say that those two input stimuli are the same, but your response to the second stimulus is lower, right? What do you call that? It's a
Well, some people call it habituation or adaptation.
Adaptation, right?
Or response to a conditioned stimuli. It really depends on which brand of neuroscience you're in.
That's right. There's all kinds of different people looking at it in different ways, but the bottom line here is that your brain is able to say, I saw the stimulus before real recently, and I'm going to pay a little bit less attention to it. I'm going to respond to it a little bit less. Is it fair to say that?
Sure.
So what Mark is asking is, if you don't see that, what does it tell you? If you don't see the brain, if you don't see the central nervous system able to adapt
to its environment,
what are you dealing with? You go ahead and tell them. I don't want to steal your thunder. I know the
schizophrenics
answer.
look like this.
Yeah.
And then people that lose their adaptation ability, and you can do all sorts of, there's lots of drugs that can lead you to that kind of response. Now you don't have to strap on an EEG and all that kind of stuff, and It used to be I guess they have dry electrodes now. I used to have you paste and all that,
Right.
shave the head and everything else to get decent resolution and put it in the right spot. EEG's gotten a lot better over the years, but still, because our hammer is sensory perception, and we see that as much higher resolution, and actually it is much higher resolution than what you can do with this because it's sensory perception.
Right.
The absolute most resolved signal is what you actually feel.
Right.
And the difference is we actually measure
The right answer is none of these really do a good job.
Yeah.
Actually at figuring out something that's happening dynamically in the brain that fast, fractions of a second. And so the focus of what we've been doing with the brain gauge is precisely to be able to see this kind of adaptation and these kinds of changes on the fly, like really fast, as your brain processes a signal and then tries to determine if it's seeing the same thing over and over again and whether or not it should pay attention to it or ignore it. That is a dynamic function in the brain that is extremely, I don't know if it's possible to get at by any other means. Is it even?
You can
That's
get,
non-invasive.
You can sort of get it 10% of it, 10 to 20% of it. It's back to what we talked about was it the last video? We talked about the Goldilocks thermometer.
Yeah, Goldilocks, that was the last video.
So you'll have to watch that one to catch up. But basically, dynamics is a really long story. It's there's lots of literature to read it on. Email me if you want to read any scientific papers on it. We've got papers going back to the 1980s. Yeah, I'll give you homework. That's
that's right. There's plenty to read. For those of you who still read, there's plenty to read out there. But anyway, I think that we're just about out of time. Did you have anything else to say?
We're gonna talk more about dynamics later. So
there's a lot more to say, and we can't do it within our time limit. So thanks a lot, and we'll see you later.
Bye-bye.